Good morning, students of standard 12. Today we are going to study topic linkage and crossing over from chapter 5, principles of inheritance and variation. Linkage. It is the process in which genes located in same or different chromosomes coexist and is associated with each other. Students, it's a very important process, linkage, which shows that it is the process in which genes which are located either on the same chromosome or on the different chromosome but still coexists, that means stays together and is associated with each other. It is of two types, complete linkage and incomplete linkage. Since complete linkage is the linkage where the genes are located quite close to each other, and incomplete linkage is the type of linkage in which the genes are located quite distant from each other. The genes which are present on the two non-sister chromatids are non-linked genes. Students, the genes which are present on the two non-sister chromatids are not linked genes. So students, here we can notice that these are the two different types of chromosomes. So that is why they will be called as non-sister chromatids. So students, these two are the non-sister chromatids, whereas these two are the sister chromatids. So students, here if the genes remain present, gene 1 and gene 2, and if they are present on the same sister chromatid, then they will be called as linked genes. But students, here also gene 1 is present on this chromosome and gene 2 is present on the another non-sister chromatid of the another chromosome, then that will be called as non-linked genes. Students, here also we can notice that the genes are located on the same sister chromatid, but the distance between the gene is more in comparison to the distance in between the genes here in this chromosome. That is why here the genes are linked, whereas here the genes are not linked. So students, this helps us to develop the concept that the genes which will be close to each other will be linked, and if the genes are present on the non-sister chromatids, then they will not be linked. The genes which are present close to each other in a sister chromatid, so more linkage. Students, this point also we have studied in the last slide, that the genes, if they will be remaining quite close to each other, then they will show more linkage. So students, the another point which clarifies this point is that linked genes do not assort independently, that is, the genes which are showing linkage don't separate from each other. So students, here also you can notice the point which is there in the slide, that genes located on the same chromosome are called as syngeneic genes, and syngeneic genes remain so close together that their alleles cannot assort independently and are called as linked genes. That is, the genes, if they are staying very close to each other, then it is very difficult for the genes to get separated. So students, here also if you will notice, the gene A with green and the other gene with red. Students, here both the genes are linked. So that is why these genes will not be able to be separated from each other. In the same way, gene B and A will be linked in such a way that they will not be able to get separated from each other. Crossing over. It is a process in which a section of non-sister chromatids exchange their segments. So students, the concept of crossing over, which is actually a process in which section of non-sister chromatids exchange their segments, can be understood with the help of the picture which is displayed here. Students, here we can clearly notice that there are two different chromosomes and both are non-sister chromatids. So students, when the non-sister chromatids exchange their section, so students, here you can notice that the chromosomes are exchanging their segments and both the chromosomes are non-sister chromatin. One is blue and the other is red. So because both of these two chromosomes are non-sister chromatid and they are exchanging their segments, there is a formation of chiasma. Students, the exact point where the chromosomes, particularly non-sister chromosomes, exchange their segments, that point is called as chiasma. Single crossing over. It is a type of crossing over in which only one chiasmata is produced. So students, here we can notice that there are two non-sister chromatids. One is red and the other is grey. Both of these two non-sister chromatids are exchanging their segments, which you can see, and this exchange of the segment, which you can see, the grey started having the red segment and the red started having the grey segment, is called as the crossing over, and the exact place where the exchange of segments takes place, that is called as chiasmata. So students, here only one chiasmata is produced, only one chiasmata is produced. So that is why it will be called as single crossing over. Double crossing over. It is the crossing over in which two chiasmata is formed. Students, here you can notice the two different chromosomes, and both of these two chromosomes can be called as non-sister chromosomes. And these non-sister chromosomes, when exchange their chromatid and there is formation of two chiasmata, then that is called as double crossing over. Students, now let's discuss an important question. What are various types of crossing over? The following are the types of crossing over: single crossing over and double crossing over. Thank you, students.